Okay, welcome back, and we're gonna go towards lecture number 3 today and now, and that's about techniques for protein extraction, a particular focus on conventional extraction methods. Okay, we have been discussing about, just to follow up on his question, why do we need ingredients? And I answered that ingredients are very important to create new foods, new flavors, new textures. And if you want to get new flavors, new textures, and new we need to extract proteins from the biomass. We need to extract proteins from seeds. We need to extract proteins from insects. We need to extract protein from microalgae. So extraction is very important. By definition, extraction is nothing but the protein is located inside and you have to remove it from inside to outside. It's a simple definition. That's called extraction. Okay? We're going to talk a little bit about different technologies that we use today to extract plant proteins or microalgal proteins or insect proteins and so on. All right? I will cover certain fundamentals now. Do you remember this structure? In primary school, that was the structure. Not primary school, maybe in the high school, we need to draw this one, and that would carry 5 marks. So shading and, I still remember my life at that time, so it was a nice picture to draw. So it's a structure of a plant cell, right? You know that plant cell, it's a lot of organelles here, right? I just want to cover again, I don't want to teach you as a primary school or high school teacher here, but you remember that plant cell are covered by this wall. What do we call that one? Cell wall, right? It's an important part of plant cell because that will stabilize the structure of the plants and the cell. Otherwise, the cell will blast, and it's an architect that it is a wall that maintain the structure of plant cells, right? Animal cell does not have a cell wall, just to refresh your memory. And below the cell wall, you have what is that membrane called? Plasma membrane. Usually it is weak and easy to damage, but cell wall is very hard and difficult to damage. Keep that in mind, okay? And this is inside, we have cytoplasm and other organelles and so on. Just remember that there is something called ER, endoplasmic reticulum, okay? Endoplasmic reticulum is the organelle that produces proteins in most cases, okay? The rough endoplasmic reticulum is the one that produces proteins. Just keep that in your mind for now, okay? And there is something called vacuoles here, you see? The big open space, sometimes filled with gas and sometimes with something, and where some things are stored. Remember these two structures, okay? Now the proteins may be somewhere inside the cells here, next to the endoplasmic reticulum and maybe inside in the vacuole, okay? And now let's see, this is a structure of, this is plant, and this is our microalgae, okay? Microalgae are very similar to plant but not the same. They are also sometimes, most of the microalgae except cyanobacteria are eukaryotic species. They have a true nucleus, and they also have a cell wall here. Can you see? Okay. Then they have this chloroplast, and then there is oil bodies, and these proteins are located here. So, take-home message from this slide is, microalgae and plants are surrounded by a very strong cell wall. Can you remember? And proteins are present inside the cell wall or inside the cell. Now, the proteins are actually surrounded by a strong cell wall. Can you just remember that? Keep that in your brain for now. Now, I'll show a little bit about the cell wall and its architecture. Cell wall is actually very strong because it has a very complex network of certain polysaccharides and sometimes some proteins, right? Plant cell wall is made up of cellulose, the most abundant biopolymer in the, it's a lot of cellulose, right? And it also contains hemicellulose, right? And sometimes it also contains pectin, right? These are the 3 polysaccharides that the plant cell wall is made up of. They are interlinked, cross-linked, and that gives a very strong, stable structure to the plant cell. That's why plants are so hard, because of this cellulose architecture. Make sense? Good. Now, if you look into the one example of a microalgal cell wall, it's very complex. It varies from species to species, family to family. Just to give a generic cell wall structure here, they also contain cellulose, okay? They sometimes also contains pectin and other things. But sometimes they also contain alginates, other kind of polysaccharides. So microalgal cell walls are still more complex compared to plant cell wall. And sometimes they're too rigid. That means very difficult to break. Right? Now, in the previous slide, we realized that the cell wall is present and the protein is inside the cell wall. Now I'm telling about cell wall is very complex and difficult to break down. Okay. Now, let's see the human digestion system. Now I take a microalgae solution. Okay. I tell that, okay, now I have created a food, contains a lot of proteins. This is my microalgae. And I tell them it is a protein content is 50%. Okay. And I'll give this person a glass of microalgae solution. And I sell that it contains 50% protein. And this solution is very rich in protein. Now you drink, you become healthy. So this person drinks this microalgae solution, right? Now let's take this microalgae as an example. The microalgae goes inside and it comes to your stomach, right? In the stomach, and before coming there is an salivary enzymes. In the stomach we have this proteases, pepsin and some lipases that digest the lipids, right? And when does the digestion occur according to you? How the digestion occurs? This is a breakdown, right? Now, you have studied in your biochemistry classes lock and key model, right? Enzymes always likes a substrate and the reaction only happens when they bind, right? You have an enzyme and you have a substrate. They would need to contact each other, then only the reaction happens. Do you agree? Now, the microalgae is coming inside your stomach. This is your stomach and the microalgae, and the protein is inside, right? Enzymes are where? Outside, in the stomach. Now, the proteins will be digested if and only if these enzymes attack the proteins. Do you agree? Now, there is a problem. What is the problem? We have a cell wall is a problem. It is acting as a barrier. All right. Now, the enzymes actually cannot penetrate through the cell wall to access the protein inside the cell. Now, we have a digestibility problem. Do you agree? Please note that plant cell wall is not permeable to most of the gastric proteases. Most of the plant, I'm not telling all, but most of the plant cell wall are not permeable to gastric proteases, lipases and amylases. So they cannot go inside because the pore size of the cell wall is very small compared to the size of the enzyme. And there is no active transporter to take them inside. Right? But sometimes they go inside in rare cases. But in seed proteins, for example, it's very difficult for the enzymes to go outside to inside and then hydrolyze. Right? Now, how do we solve this problem? We need to break the cell wall, right? That I answered your question again. We can't eat the whole biomasses in most cases because of the cell wall. Only animals can do this one. For example, we have cow, for example. They have a special digestive system. That's why they eat grass and they can digest it through fermentation and so on. But we can't do that. We can't eat grass because it's a lot of cellulose. We don't have an enzyme called cellulase in our stomach to break down the cellulose. Our stomach is containing only amylases, proteases, no cellulases. So we need to somehow open and then you give or chew it properly, break down the cell, and then you can make the protein accessible or digestible. Does that make sense? And there, the processing becomes very important. I cannot feed them the whole microalgae here. Maybe I need to unlock the cell and feed them only the protein. Does that make sense? And where the extraction becomes super important. The conclusion is very simple. Plant cells, animals, microalgal cells are bound by a strong cell wall. Proteins are inside. It's sometimes very difficult to digest. So we need to unlock, we need to open the cell wall, break the cell wall to make the protein accessible to the body. Right? And there it comes, the extraction becomes super important. And that also leads to the ingredient production. Any question in this part?
So does it mean that we are consuming these cereals, they are not digestible at all?
Yeah, no, I don't say that one because we cook it, we germinate it, and then we help damaging the cell wall. I'll come back to that one. Yeah, raw, no. Less digestible, I'm sure. Yeah. You need to ferment it, you need to germinate it, you have to cook it, you have to boil it, and all this process will damage cell wall, makes the protein more bioaccessible. That's another story. Okay? But raw, no. Raw ingredients, no, because of this problem.
So that's also applicable to animals also.
Exactly. Yeah.
So, yeah. Thanks.
Now, I'll talk a little bit more into exactly where the proteins are located in seed storage proteins. Now, I taught you yesterday that soybean is an important source of soy protein isolates, right? Now, this is the structure of soy seed, and if you cut it into half and you get this cotyledons, for example, okay? And then we have this place where the proteins are located. Now, this is a microscopic image. The proteins are located in a specialized structure called protein bodies, PB, okay? Protein bodies, okay? And it's not spread throughout the structure. It's present in specialized locations as a protein bodies, okay? Now, we need to bring them out to get them as a protein ingredient, okay? Now, I'll show a little bit more about how exactly the protein synthesis happens in the cells. Now, this is inside the seed in one of the structures you see. Basically, in one of the cells, you have this endoplasmic reticulum. I showed in the plant cell, same thing, okay? And that synthesizes the proteins, okay? Then immediately after synthesis of the proteins, there may be this tertiary structure and quaternary structures can happen. That means the folding and everything happens next to the endoplasmic reticulum, right? And then the proteins, as protein bodies, they travel and then they will be located in a special structure called protein storage vacuoles. There will be small vacuoles here, and the proteins are actually stored inside the vacuoles, okay? The endoplasmic reticulum synthesizes the proteins, and it's transported and located in the vacuoles. And everything is happening inside the seeds now, right? Now, remember, the seed has seed coat and a lot of other barriers also here. Now, we need to extract actually these proteins out. Okay, we need to extract these proteins out, and to extract these proteins out of the seed, we need extraction technologies. Extraction technologies. Okay, so just to take home message is that remember the seed and the proteins are not spread everywhere. It's present in a specialized places called vacuoles. And inside the vacuoles, they are present as the protein bodies. And to get them out of the cells, you need to damage the seed and get them out. Okay. So, I can skip this slide. It's not so important. In case of microalgae, they don't have a specialized vacuoles to store the proteins compared to plants. Okay. They say that they're present in the chloroplast. This is chloroplast. So, the proteins are located in chloroplast. Chloroplast, you know, right? The light harvesting pigment that helps in photosynthesis, okay? And some people also say they are present in cytoplasm. I could not find a very good literature to prove that one, but it's expected that they don't have a specialized protein bodies in the microalgal cell. They're spread in the cytoplasm or in the chloroplast, especially in the in certain location of the chloroplast. What is it called? Pyramid or something like that. Clear? So in plants, we have special protein bodies. In microalgae and other places, just spread across. But cell wall is always present, okay? Then we need to extract protein from inside to outside. Now, I'm going to show you how exactly we can extract protein from inside to outside. Protein is inside and protein is now outside using different extraction technologies. So, that can be divided into two classifications, conventional extraction methods and novel or emerging extraction methods. In case of conventional extraction methods, we have a dry fractionation, wet fractionation. In wet fractionation, we have salt extraction and alkali extraction. In novel, we have a lot of things, but I'm only covering 3 of them today: ultrasound, pulsed electric field, and enzymatic. It's just to show you classification. I'm not covering 100%, but I'm covering 80 most important ones today. Is it clear? So, idea is to extract protein from inside to outside using different technologies. Okay, each technologies have its own advantages and disadvantages. That's we're going to discuss today. What I'm going to teach you is the principles of extraction, okay, advantages and disadvantages, and some examples. That's it. You are welcome to ask me any number of questions in between. All right. Let's begin with this one, the conventional extraction today. In the conventional extraction, one of the most common or the cheapest method is dry fractionation. Okay. I really found these figures from this article because that saved me a lot of time. They are fantastic figures. So I just copy pasted here. Dry fractionation, basically, as the word says, fractionating seeds based on the dry seeds without using any solvents. No solvents are involved, just using some methods, we're just fractionating them. How do we do that? Just an example. You take pea here, okay? You take not fresh one, the dry pea, okay? And put it into the mill here. You have seen this mill, right? What we call to get the flour. It's very common in India. So mills are used to get the flours, right? Basically, you mill them and you get the flour here. Okay? Once you get the flour is now basically mixture of proteins, carbohydrates, small amount of fat, and lot of other things, anti-nutrients and everything. Do you agree? There's no separation has taken place here. It's mixture of everything. Just think about wheat flour. It contains everything. But you are welcome to do the dehulling first. Just remove the seed coat first using the dehulling machine. Otherwise, usually the hull of the seed contains a lot of phenolic compounds that's really nasty. So you first do the dehulling, and you're also welcome to remove the oil fraction using some solvent extraction or boiling and then redrying. You can also remove that one. The idea is take the dry seed and then mill it, you get the flour. Once you get the flour, you do the air classification. That's a technology, and that will help you to separate protein fraction from the fiber or the starch fraction. Okay? So we have the flour, and then we do air classification, and then you get the protein fraction and carbohydrate fraction. Now I'm going to show you what exactly we do it in the air classification. Basically, we supply this flour here, the what you got it from the milling, okay? And then there is actually a wheel that continuously rotates, creating the centrifugal force, okay? A wheel continuously rotates, creating the centrifugal force, and we supply air in this direction, okay? It's just a chamber, you're getting the feed, and there is something rotating here, then you supply air in this direction. Now what happens is separation takes place because every particle has its own density. Do you agree? Its own size and its own density. Proteins are less denser than fibers and starch. Proteins are less denser than carbs, mean fibers and starch. Okay? Now basically purely on the density based separation, you get a fine fraction which contains protein, and coarse fraction it usually contains carbs, which is starch and fibers. Okay? It's the most simple way of separating proteins because proteins are less denser than the carbs. Is it clear? Okay. That's how you get the protein fraction and starch fraction separated from, especially from cereals. From legumes, it's a little bit challenging. Why? Because it has fat. When you have fat, what happens? It damages this principle because these oily matrices changes the density of the proteins and carbohydrates because of the interaction. So it's always recommended that you remove the fat before you do the air classification to get the best separation. Okay? Any question in the air classification? Yeah, as fine as possible. But if you really want to talk about the size, I don't recall exactly. I think the fine particles here, what you get, the proteins around 303 micrometer, and coarse fraction, I think the starch fraction will around above 5 micrometer. That's the size we are separating. So you mill as fine as possible. That's very important. There's a lot of pin mills and there's a lot of milling technology today. So I'm not talking about extremely fine one, but it's actually still a coarse fractionation where it's possible to mill as fine as possible. The property is purely on the density and the particle size.
Just like frictional properties, density is one, like gravimetric, frictional, mechanical property. Which property we are focusing in this separation?
Density. Purely on the particle size and density.
So, but if we increase this air in inlet, velocity of air inlet, it will be difficult to separate the size.
That's exactly that I did not cover here. Basically, to control the separation, you need to control the velocity of this wheel here and the force of the air. These are the two controlling parameters. that will change how you're getting this fraction and this fraction. I will talk a little bit about challenges as you may ask this question. Basically, I don't claim that you'll 100% get proteins here, 100% you get carbohydrate. That's not possible. It's always this contains small amount of carbohydrate, this fraction contains small amount of carbohydrate, and this fraction contains small amount of protein. This is the biggest challenge because I'm not talking about pure based on the density. It's an impure, crude way of separation. It's one of the disadvantages of this particular method.
So is there any role of aerodynamic properties of particles with this operation?
Could be. Probably, I don't know. That could be. Maybe it's not my area of expertise. I can't answer this question because I have no idea about aerodynamic properties of the particles. I never studied it myself and never read it myself with intention. There could be. But what I know that this separates purely based on density and the particle size.
So because whenever we separate anything with respect to velocity of air or with respect to air, aerodynamic properties are important to separate them.
Could be, yeah.
If it is a combination of size and the
density and the aerodynamic property. I fully agree with you.
The reason for the separation of protein on the basis of size.
Could be, yeah. Because, of course, I don't have this engineering background. Very difficult to give some explanation about the aerodynamics properties. You would be right. Let's read it about this point, okay? Good point.
Thank you.
But what I know is that its density is very important. Gravity, basically. It changes and then you get the fraction. Is it clear? Air classification. Then sometimes they can also use this electrostatic separation. The phenomenon is very simple. When you supply this air inlet and the feed, basically there is a collision between the particles. Okay? Particles collide each other and particles also collide to the wall of this instrument or the equipment, the barrel, right? Usually because of the collision between the particles, there will be charge transfer. And they say that the protein will get positively charged surface and the carbohydrates get, this is protein, and carbohydrates, starch, let's say, get negative charge on the surface because of the charge transfer reaction. And that will also help us to separate further. Now what they do is that when they're colliding each other, you get a charge, positive and negative. Then you have an electrodes here. Cathode is positively charged and anode is negatively charged. This is positive in this case and negative in this case. Carbs in this direction and protein in this direction. And based on the charge, they can be separated. This is another way of classifying using dry fractionation. Clear? I don't want to give a lot of explanation here. Just remember that because of the collision between the particles, the protein particles and the starch particles or the carbohydrate particles can get charge. Okay? They get a surface charge and the charge could be positive on the proteins and negative on the carbohydrates usually. And it's very difficult to say how they get the charge. People say that the transfer of electron, that the transfer of ions. I could not find any strong evidence for that one, but everybody knows that the particles are actually charged. And based on the charge, you can actually separate them into different fractions. So you get a protein fraction here and you get a carb fraction. You have a question? Yeah.
But in case of press cakes, can you do it?
It's very difficult again. Basically, what they do is that you do this hexane extraction first and remove most of the oil with the hexane extraction.
After that, it can.
And then it can be done. But it's always a challenging with basically, you need to get a kind of a flowery matrix to do the air. If you have lumps, it's always a challenge. So, yeah. It works really well with cereals, if I'm not wrong, compared to legumes. But that is also one of the disadvantages of this methods. Basically, if you really want to go with an air classification with oil-containing matrices, you need to do a pre-treatment called de-oiling. And that's a very difficult process. Easy process if you would add an hexane as a solvent. But hexane is not environmental friendly solvent, and people talk about environmental footprint with hexane. Or you have to do boiling first, then you separate oil, and then you have to dry them. The drying is very energy consuming process in whole food processing. Drying consumes a lot of energy. So considering all these things it's a bit difficult and challenging. It's not the best method in the world. However, it's really easy and energy saving method provided you have the right feed here. Any questions? Yeah.
How we can extract these as proteins are macromolecules? How we can separate them from the micromolecules like other components which are with the grains or which are attached with the flowers.
This is a good question.
The percentage of purity. What
Yes.
is the purity?
I will. That is one slide. I clearly show how much percentage purity you get. It's in the end of the slide. We are not talking about 90% purity and 80% purity, 100% purity here. No. Air classification cannot get 90% pure protein. Impossible. This protein you get here is not 90%. Maybe 40%, maybe 50%, maybe 60%, depending on the type of the flower you have. And the impurities always anti-nutrients and phenolics and carbohydrates, everything will be present here. The remaining things. So it's not the purification method, it's the fractionation method. Does that make sense? It's not a purification method, it's a fractionation method. If you really want to do 90%, you need to purify it again with other different technologies, which we will cover later. Make sense? It's just to separate, to get a rough fraction. Now, can you tell me if this protein is useful somewhere?
Yeah, we can. So it's rich in.
Can you think about a food product where I can use this for this fraction?
Yeah, those foods which are good, which need a structure.
Yeah,
we can add this protein resource.
Example? Do you have an example? There is a person you have. Fortified food, for example, you can use it in, let's say, baking, for example. You don't really need ultra-pure proteins for baking, right? And there, basically, these kind of fractions can be used without any problem because they're the sensorial properties. I'm not telling that you have to use 100% of this protein. They usually use 50, 20% is replaced. The wheat is replaced by these alternative proteins. Okay? So this is not one of the advantages. Very fast, less energy consuming. Disadvantage is that it does not produce pure proteins. You can't get protein isolates by air classification. Am I clear? Good. I need to go a little bit quickly. I already covered the advantages and disadvantages. But do you think any other disadvantage here that I did not cover? Any advantage or any disadvantage?
It is easy method. There is no use of solvents. There will be no impurity like a chemical impurity.
Exactly.
There are no environmental hazards of this method. And if we come to disadvantage, purity will be very less.
Yeah.
Anti-nutrients may be present.
Yeah.
And other non-nitrogenous components may also be attached with it.
Yes. So he has covered everything now. I don't want to explain this again. Is it clear? So, no solvents have been used unless you do de-oiling in the prior. Sometimes you need to use it. Then it becomes expensive and less environmental friendly. Easy because you need a specialized person to do these things. Somebody will mill it, somebody will add it, somebody will classify it. It's very easy. You don't need a PhD for that. It can be done by anybody in principle. But the biggest challenge is the purity is very low. We're talking about 40, 50, 60% pure. Anti-nutrients will be very high because they always come together with this fine fraction because they are also less denser. And impurity will be very, very high because if you say 60% pure, the 40% could be impurities for us. Remember that if you have more carbohydrates, you will compromise the functional properties of the proteins because you can't use it in every food systems. All right? Now a little bit moving on to the salt extraction. This is also one of the common approaches to extract proteins. What we do here is that let's take pea again as an example. You mill it and you get the pea flour. Okay? Then the flour is suspended in a solution containing salt. It could be sodium chloride. Right? And if you go back to my previous slide, previous lecture, I taught you some basics. You remember? Some proteins like salt-soluble proteins. Example, globulins. And some proteins like water, albumins. Some protein likes alcohol, prolamines. So every protein has some tendency to dissolve in some sort of solvents. Basically, that principle is used here to extract proteins. Nothing difficult here. Some proteins like salt, they are solubilized. And some protein that does not like salt, they will be precipitated, right? Then you centrifuge, get a 2 fraction salt-soluble protein and salt-insoluble proteins, dry it and use it. That's the principle of salt extraction. Very easy, right? Of course, it has its own challenges, I'll tell you later, but this is the basic principle. Clear? purely depending on the solubility of protein in the salt solution. You can actually change the concentration of salt and you can selectively precipitate different kind of protein. That's also possible. That's the advantage of this technique. That I'll explain over here. Now, this is actually a figure I copied from this particular research article. I think this is the protein from yeast, if I'm not wrong. You can see this in the y-axis. This is y-axis, solubility. X-axis, the concentration of sodium chloride. The solubility was here, increasing, and then it decreased. Right? The protein solubility increased, got the highest at when the concentration of sodium chloride is 1 molar, and then it started decreasing. Can anyone explain what's exactly happening here? Based on the charge, isoelectric point and charge, but we are not changing the pH here. Addition of OH group.
H positive. In alkaline conditions, we are adding charge to it.
But I'm not changing the pH at all here.
We are changing the concentration. The more H2 positive will be there, so there will be...
Okay, let's avoid this discussion. I tell you that the concentration of H plus and OH minus in this system is fixed. Let's say we are doing the extraction at pH 5.5, okay, where the protein is having one set of solubility. Just to avoid this discussion, what would be the other problem? Electrostatic attraction and repulsion is playing a role. That's a good point. So, sodium chloride has a charge, right? Na plus and Cl minus. Remember, as soon as you take a glass of water, take salt, table salt, put it into and then you shake it. Then the sodium chloride dissolves in water. When it dissolves in water, it gets Na plus and Cl minus. Okay. Now we have a beaker, okay, containing protein. This is protein, and it contains Na plus and Cl minus here, right? Now, going back to my previous lecture, I told you proteins are charged. Did I say that one? Because of the amino acid, and somebody told it's amphoteric, right? Proteins can have positive charge, can have negative charge, and so on their surface, depending on the amino acid residues and the pH. Lysine can become positively charged, glutamic acid can become negatively charged, arginine can become positively charged, aspartic acid can become negatively charged. So they change their charging depending on the amino acid side chain and the pH of the solution. Now, proteins are charged, right? Now, you have another protein here, and that is also charged, imagine. Because of the charge, they either attract or they repel, right? At isoelectric point, they completely attract each other, they precipitate, because the net charge is 0, right? At the non-isoelectric point conditions, they are having different charges. They don't not necessarily attract, not necessarily repel. It varies. Is it clear? Now, I add sodium chloride to this solution and it contains Na plus and Cl minus, right? And what this sodium chloride does, you have a sodium contains positively charged and you have negatively charged here. Can you see? So, that will shield here. And you have a positive charge on the surface and you get a chloride ion. Basically, the protein charge is shielded. Can you see here? And here also protein charge is shielded. Now, since the charge of the proteins are shielded, protein-protein interaction is reduced. So, protein interaction is reduced because of NaCl. Can you see? Because of the NaCl, the protein-protein interaction is reduced. Now, the protein don't love each other. They just want to go away. But they start liking the water molecules. Okay? Now, the protein will be completely hydrated. Hydration of protein is nothing but solubility of protein. Now, the protein will directly go into water. Is it clear? And that's what exactly happening in this region. Okay? In this region, protein charge will be shielded by the positive and negative ions of the salt. So, the protein-protein interaction is reduced. Protein-water interaction is increased. Increasing protein-water interaction is nothing but solubility. So, in this region, the solubility of the protein is increasing because of this reason. Okay? I should have not written in this part because my photo will come here. So, but is it clear? All right. Now, we understood this part of the curve. Can someone explain what exactly happening here? Highest solubility. It's a critical point where we have a sufficient amount of NaCl which will completely shield excessive charge on the protein. Then the protein will really love water molecule and then solubilizes. Is it clear? Now, there is again half of the answer for this question. There is a second half of this answer question. Can somebody explain what exactly happening here? Why the solubility is decreasing? Charge is again increasing, but what is the problem? What it will cause? Why you get a protein precipitation? Why protein is insoluble now? Increase in the total mass is not necessarily true. Because of the repulsion, what is being repelled? Can anybody think why the solubility is decreasing here? Okay, I give a hint. Solubility is because of protein-water interaction. Okay? Protein should love water if it wants to get solubilized in water, right? Solubility decreases when this interaction goes down. Now think about how this interaction is broken here in this particular part of the curve. Why we have a reduced protein-water interaction and increased protein-protein interaction in this region? Because of the saturation of the NaCl. That's the right answer. How and why? What exactly is doing?
More salt will be engaged with water.
Exactly. Now, NaCl is start loving water rather than the protein, right? You are adding more and more NaCl, then that another effect comes into play. That's called hydration of salt ions. Now, NaCl is positively charged, right? Chloride ion is negatively charged, right? Of course, Na also likes protein. It also likes water because you know that the water has two charges, right? You studied in your chemistry class. A delta positive and delta negative, right? Because of this oxygen molecule, right? Now, Na plus likes the negative charge of the water molecule. So, water molecule starts surrounding the Na like this one. Okay? Basically, the sodium ions and chloride ions are hydrated now, more and more. Now, these ions start pulling the waters towards them. And now the water is no longer available for the protein. And then protein cannot hydrate it. And they hate water molecule because they're not having water molecule any longer. They start loving each other and they precipitate. Is it clear? Okay. And this is the reason why the salt how it is affecting the solubility. And this region is called salting in, and this is called salting out. This is the two phenomena you should remember if you're studying solubility in food science and technology. Salting in and salting out. Is it clear? I have a nice explanation here again. In this region, you have a salting in, and in this region, you have salting out. In this region, what is happening? Protein-protein interaction is reducing and protein-water interaction is increasing. This is protein, this is water. Okay? And basically, it's because sodium ions are shielding the charges on the protein. They help protein-water interaction, not protein-protein interaction. But if you cross the threshold, then there is a more salt ions and these ions will hydrate. They pull the water molecules towards them. So, protein-protein interaction increases and protein-water interaction decreases and the protein can precipitate. The solubility can decrease. And this is a phenomenon you can use to improve the solubility of your plant proteins. He mentioned plant proteins are less soluble. You can make them more soluble by using salt at right concentration. That varies from protein to protein. Usually 1 molar sodium chloride is used. In most cases, you'll study a literature article, they use 1 molar sodium chloride because that is often helps the solubility. Should I explain it again? Clear? This diagram is clear, right? I'm sorry, I made a big mess here, but hope you understood. Clear? All right. And there is something called Hofmeister series. It's a very well-known series to, just ignore this one. It's a very well-known series to show the which kind of salt you can use. For example, if you really want to do the salting out, use this kind of ions. If you really want to do salting in, you can use this kind of ion. But in food science, we use sodium chloride and usually sodium chloride because sodium chloride is food grade. You can also use ammonium sulfate to precipitate the protein. I think ammonium sulfate is also food grade. You can buy them. Is it clear? Salt extraction. All right, now can you tell me some advantages and disadvantages here? Yes, we basically have to do it either salting in and salting out, or both of them. It depends on what do you want. Basically, protein will start separating here depending on the salt concentration. Some will precipitate here and some will precipitate here and some will precipitate here. And then you basically try adjusting the salt ionic strength of the solution. Then you can selectively start precipitating. That's possible. But basically, you need to do both salting and salting out. But you can only use salting in. That is also possible. How? Let's look here. If you do only salting in here, basically all the salt soluble will be in the solution, and whichever completely hates the salt will just pellet off here. And that's the reason you centrifuge and you just get rid of them. If you do the salting out, first you do the centrifugation, remove the insoluble part, and then you do salting out and you get the fraction with that is dissolved here. It depends upon what you want to do. So what would be the biggest disadvantage of this salt extraction? My answer is already in my question. Presence of salt is the biggest disadvantage. After doing all these things, I said that, okay, you go for spray drying or freeze drying here, right? Can you go directly? No, because your supernatant has a lot of salt now. Basically, what you get here in the dry fraction is basically protein plus some anti-nutrient and other things, not 100% pure again here, plus salt. Nobody likes sodium chloride today, right? BP, blood pressure. So we don't want to have a lot of salt. Of course, the taste is a problem. Another thing is we really want to get rid of almost all sodium ions. It's not good in the food system, right? So you need to do one more experiment here. What is called? Dialysis, an extra step to get rid of salt. Unless you do salting out. But again, there is a salt. It's not an easy process. So you need to do dialysis or membrane filtration to get rid of salt here. And that's an added experiment to do. That's one of the biggest disadvantage. Easiest is advantage is, what's the advantage? Easily available. Sodium chloride is one of the cheapest thing to buy in the market, right? The cheapest even today is sodium chloride. Salt is cheaper. You don't need ultra pure salt here. Normal salt will do the job, right? So this is the advantage, this is the disadvantage. But please note that this method again doesn't give you the ultra pure protein. The protein concentration can be as high as 80% in some cases, depending on your raw material. Depending on your raw material, you can get the high purity protein. It's usually better than the dry fractionation. All right. So people often combine both dry fractionation and wet extraction together. It's a win-win approach because in the dry fractionation, you get the fine fraction rich in proteins, right? Use this fraction in the salt extraction. Basically, here you get around 60% purity, and do the salt extraction, the purity can become maybe 80% here. If you only start without this one, the purity can be 70% here. So basically, you can combine two techniques together to get little bit higher purity protein, and that's often done actually in the industries. They don't do one extraction, they often combine two extraction depending on what do you need at the end of the day. Am I clear? So this is often used, combination of dry fractionation and this one. People call it as mild fractionation, gentle fractionation, and so on for this technique. Another important technique today that we're going to discuss is alkali extraction. I'm pretty sure you have read about this a lot. And this is one of the most common used technique to get plant proteins, microalgal proteins, insect proteins, and so on. Okay? Alkali extraction, industrially followed, can be scaled up, extremely well investigated, can be produced things in very bulk, high quantities. Okay, so this is the most used approaches to produce plant protein isolates today. Okay, compared to salt and compared to other extraction method I have used. Now, let's talk a little bit about alkali extraction. The process starts exactly the same thing. You get the flour of given matrix. It could be pea, it could be soy, whatever it is. You're welcome to do de-oiling. Please note that I don't mention de-oiling here. You're welcome to do de-oiling if you're really necessary, especially for oil crops, oil seeds, okay? And then basically you dissolve them in a solution containing sodium hydroxide or potassium hydroxide. These are often used. Sometimes people also use sodium bicarbonate. Sodium hydroxide can be food grade, right? we all know something that protein solubility changes depending on the pH, right? This is the curve I taught you last time, the basics of protein. I taught you this curve, right? Protein has a very good solubility at particular pH, and the solubility drops, solubility completely lost here. What is that point called? PI, isoelectric point. And then if you increase the pH, the solubility increases, right? Now we have two regions where we have the high solubility, right? This is the basic principle of alkali extraction. High solubility and low solubility. Both these aspects are used, okay? Usually, alkali condition here, the solubility is actually maximum. Can you see? Compared to acid condition, alkali condition has a higher solubility. So they use this phenomenon, they exploit this phenomenon. They adjust the pH between 8 to 11, you often, right? And when you adjust the pH 8 to 11, how do I get rid of this one? When you adjust the pH around 8 to 11, the most of the protein will be solubilized here, okay? Then what they do here, the centrifuge here, then the pellet contains insoluble fractions. It could be fiber, it could be hull matrix, it will be starch and whatnot. All this insoluble, alkali insoluble fraction will be precipitated off. So you remove them by centrifugation, right? And then you have a supernatant here. What does that contain? Soluble proteins, okay? Then you remember the same curve here. The protein is soluble in this region, but the protein is insoluble in this region. Now what we can do? Precipitate by adding acid or HCl here, right? You add the HCl, bring the protein to its isoelectric point, and the protein will be precipitated. So now you get the tube containing protein precipitate and some water HCl here, right? Basically it's water because it's neutralized. Am I clear? So basically you precipitate the protein here and again centrifuged, you got the protein fraction. Is it clear? We are purely using the solubility curve of the protein to extract the protein. We use both isoelectric point concept and the solubility concept here. Since usually alkali conditions is more preferable because that more solubility means what? More yield. Do you agree? You throw less here. More solubility means more yield. You throw less in this particular part. And that's the reason why people don't use acid extraction. You're welcome to use acid extraction. I'm not telling that you can't use it. You can use it, but the yield often lower compared to alkali extraction. All right? And then you precipitate them again by adjusting the pH to PI. Then you get the protein out. Exactly. There's a lot of disadvantages here. That's on my next slides. I'll get back to exactly color problem and sorting. Good question. I'll get back to you in detail. Oh, no, you can always use this pH is equal to the pH and PI equation. What is it called? Anderson-Hasselbalch equation, right? That is showing basically when you have an pH becomes PI when then the acid form and the base form becomes equal. Then the net charge becomes 0. There's an Hasselbalch-Handels equation. You can Google it. You'll find it. There's an equation. Basically protein has properties called denaturation. Basically, I remember tertiary structure and quaternary structure, they're beautifully folded. And that is actually governed by hydrogen bonding and hydrophobic interaction in addition to disulfide bond. Because of the alkali condition, they can open up because you can even break the disulfide bond in alkali condition. Usually, proteins have two properties. Sometimes denaturation is reversible. Sometimes they're completely irreversible, depending on to what extent you're damaging. As soon as you remove the salt or the base and acid here, they can refold to a certain extent, but sometimes you cannot. That's a balance. So, remember one thing is that we do not always need native protein structure in food applications. We do not need it always. It's okay to use denatured protein for food application. Then application changes. Sometimes we really have to denature protein to improve the water holding capacity, oil holding capacity. It depends on the application. So, food processing will always denature protein to a certain degree. It's difficult to maintain the nativity. The nativity is important when you want to use it them as an enzyme, because then the quaternary structure and everything becomes super important. But proteins is not necessarily if you want to use it for the food ingredients. Yeah.
One more point I want to just have a discussion on this, that during my PhD research work, I isolated the protein using acetone precipitation, and then further my supervisor said that you have to maintain the functionality because we need to go for the correlation with the product quality at the end. So I went for dialysis after studying the literature that by doing the dialysis against the acetic acid, oxidation of the bonds will occur during the dialysis process, and there may be some maintenance of the functional property of that protein which I extracted, and there will be further more purification also, because dialysis will remove the minerals in case they are being attached with the proteins. So what is the possible chemistry behind this? Because you said that your expertise is the protein oxidation.
So it's basically like, let's say that this is a protein which is three-dimensionally folded, and then you have a disulfide bonds here like this, right? And because of XYZ treatment, then they become like this or they become like this and so on, right? Basically, you denature proteins, the three-dimensionally folded will become open up. Now, this one has one type of functionality and this one has another type of functionality. It depends what do you actually need in food application. It's not that, okay, native is the best in the world. Not necessarily true. Maybe denature will give you the more functionality. So don't go with the myth that we need a native, but we need the native protein to understand the chemistry. I fully agree. We're trying to get them as gentle as possible, as denatured, less denatured as possible, but just to understand the functionality. But we don't claim that, okay, denaturation will completely remove their functionality. It's not true. It depends upon what exactly you need at the end of the day. Maybe denatured is more digestible than the native one. You never know. So, but coming back to the chemistry point of view, basically, I have the slides later. If you have more hydroxyl ion, basically you cleave this bond and you will denature it. And if you have more acid conditions, you protonate them and it will repel each other and you help, you break the hydrogen bonding, and then you open up the structure and then it will easily cleave this bond. There is a lot of chemistry going on. So one thing to keep in mind is that probably native is good, but may not be very functional. Denatured is maybe better. Maybe it's bad also, depending on the protein and the condition. But the problem here is that to denature, you add a lot of ingredients, NaCl, NaOH, and all these things have more effect, which I'll try to cover. Okay? Yeah.
Sir, why we need to extract protein? Suppose we are having 60% of protein percentage in. Why not we go for the other component to extract, like carbohydrates are present there? We extract 40%, we will get 100% of protein. Why we need to extract protein only? Why not the other components, which are minor compared to protein?
That's exactly what's happening in this particular step. Basically, you're extracting protein in this. For example, you get a protein supernatant here. The pellet, what you get here is actually containing the minor, the carbohydrates, fibers, and so on. But the supernatant again contains polyphenols and all the soluble fractions.
Suppose our sample contains 60% in trifractionation. It contains 60% of protein. There is 40% are another component like fibers, any other polysaccharides. So why we do not go for the polysaccharides to extract? So we will get 100% of protein.
How do you extract polysaccharides?
There might be methods. There is one method we can go for, like enzymatic.
That's again extraction. So whatever you say is again extraction. Is the focus point is different. What you're extracting? I'm extracting proteins. Basically, you're also extracting carbohydrates. With me, all the fibers are coming here. So I can tell my fractionation for carbohydrate extraction is the same thing. It's basically the focus point is different. Basically, the process is different. But at the end of the day, it's extraction. I don't care what method you're using to get what. But at the end of the day, you're fractionating in different fractions. One fraction will have protein, other fraction will have carbohydrate, one fraction will have polyphenols. It's again extraction.
Yes. So it's simply this a question. There is a minor percentage of another impurities.
I'll get back to this point. The minor is actually a bad thing to use here. I can give you one example. Less than 1% of polyphenol on a protein can change the protein quality completely. It can make protein very brown, it can make protein bitter. Only less than 1% I'm talking about. So you have 99% pure protein. I can say, oh, I have a 99% protein, but nobody can use it. It's bitter. Can you buy that? No. That's the reason why fractionation and purification becomes super important. One thing is, please note that fractionation is not necessarily good. Basically, you're also up-concentrating, in some cases, unwanted things here. You're removing one thing, but you're also up-concentrating another thing here. Continuously, you're concentrating polyphenols in the supernatant. Do you agree with me? Because polyphenols don't go in the pellet. They're soluble, unless they're bound to the carbohydrate. So you're also up-concentrating. This fraction you get over here, maybe it's rich in protein, maybe rich in antinutrients also. Then you need to again purify them in other technology, membrane filtration. Yeah, you have a point here? Yeah.
I have a question, sir.
Yeah.
Actually, I have two questions.
Yeah.
First one with the extraction.
Yeah.
So after doing isoelectric precipitation,
Yeah.
There also the reaction happens and salt will be formed.
Yes, NaCl, exactly. Yeah.
And if we wash it multiple times,
Yeah.
so how sure are we that it's removed?
Yes, it's a good question. Basically, NaCl content will be there. It's actually a game, trial and error game. But there is definitely something, because usually the pH will govern the NaCl concentration. pH will overweigh, the isoelectric point will overweigh the salt concentration. So protein will precipitate. So it's possible. Yeah.
How sure are we that it is removed? That's my question.
Yeah. Again, you do the protein analysis. You take the fraction, go to the Geldar's method, CHNS analysis, chromatography, BC analysis, measure it. So that's the only way. All purely trial and error here. Once you optimize the process, and then it is industrially applied, because I know at this pH it will precipitate. I don't care about NaCl now, because still it precipitates. It all has to be optimized for individual protein, for individual matter. It's not a universal method I'm talking about. Please note that. It's a universal method, it works. But this is not a universal method, but it works basically. This is a universal thing. That is unique for each protein classes, right? Use that as a key factor to find out where do you want to precipitate. It's a trial and error. If it's new protein, yeah.
One more question, sir.
Yeah.
Actually, mine is a protein which is not much studied.
Yeah.
So I have extracted protein at pH 10 and pH 12 as well.
Yeah.
I have found my isoelectric point at 3.6.
Yes.
So when I did extraction at 10, it gave more pure protein.
Yeah.
But the emulsifying capacity was less.
Yeah.
When I extracted at 12, yield was more.
Yeah.
Purity was a little bit compromised.
Yeah.
But the emulsifying ability was much good.
Yes.
So should I attribute this to the polyphenol content?
You could be. I'll come back to that particular answer in next slide. Good question. Could be. Yes, you're right. Yeah. Shall I continue? Any other question? I think it's a fantastic discussion. I'm pretty sure in understanding what I'm teaching now. It's cool. So is it clear? Alkali extraction. Please note that it's most commonly used because it gives highest yield. Okay. I'll answer your questions along the way, and also your question about polyphenol and minor components. So this is one of the examples. It's our own study we published, I think, back in 2020. We're trying to extract proteins from hemp seed meal. Hemp, right? It's also produced in India. We can see that increasing the NaCl, potassium hydroxide concentration, we increase the yield. Okay. And if you compare the yield with the salt extraction with the alkali extraction, you can see that the yield is maximum is around 30% here. There you go up to 80% here. So usually alkali extraction produces highest yield compared to salt extraction. It's not only in this study, a number of studies prove that one. And that's the reason why people go for alkali extraction compared to salt extraction. Please note that the protein profiles here could be different, because this will extract only one set of proteins and this will extract different sets of protein. And that's the reason why your functional properties also changes depending on the pH, because the solubility varies depending on the pH, right? At pH 12, you might extract more number of proteins, more diverse protein compared to pH 10. And basically remember, plant proteins are not a single protein. It's a mixture of hundreds of proteins. Each protein will have their own isoforms, their own proteoforms. There's a small difference in them. And their solubility is different depending on the salt concentration and the pH. So your functionality, what you're measuring, not for the individual protein, it's for the mixture. So the small change in the protein proportion in the mixture can change the protein quality or functionality. That's one of the answers. So that's the reason why the protein coming from the salt extraction can have a different functionality compared to alkali extraction. Alkali extraction at different pH can have a different functionality because you're actually extracting different types of protein at different proportions. Okay? All right, and this is about the digestibility except for the, this is from, I think it's from our department, this paper. I think it's from rapeseed, right? Can I read some rapeseed? Yes, exactly. This is from the rapeseed protein. You can clearly show that the salt extraction has a lower digestibility compared to alkali extraction, something like that. So the digestibility also varies depending on the which type of extraction you're using, because sometimes some extraction will denature the protein, then the enzymes can easily access to the protein, can be more digestible. So sometimes denaturation is good, sometimes it's bad for the functionality. So it depends what is your end goal. End goal is to have the highest water holding capacity, go for salt extraction, maybe, I don't know. End goal is to have a highest digestibility, I don't know, you have to go for salt or alkali extraction. It varies from protein to protein, materials to material. It's again a trial and error method. There is no universal extraction method that gives you the highest yield, highest digestibility, highest water holding capacity. No. You tweak it, you train your protein to get the best one, what you want. Make sense? And that's the reason why the plant-based world is so complicated. It's very complicated because of this, all these things affecting the end result. Okay? Coming back to your own point here, there was a study published a few years ago. They clearly shows that the water holding capacity and oil holding capacity is different depending on the extraction you're using. If you use AF, NOH, they have a different solubility. If you use NOH together with salt, they have a different solubility. And their water holding and oil holding capacity is changing completely depending on what type of extraction you're using. It's clearly showing us that extraction actually matters a lot toward the functionality. So if you want a low water holding capacity, probably you have to use this method. If you want the highest one, probably you need to use this method, is this one. So the methods matters. The extraction method matters to get the right functionality. Please remember, we don't have to use always the highest water holding capacity ones. It depending on where do you want to use this protein. Make sense? So there is no universal method to get everything as the best one. No. Use the you optimize it, you find out it and find the best one. There's a lot of room for exploratory research. All right, can you talk a little bit about the disadvantages here? One of the disadvantages you asked about, okay, what about the purity and what about the color and something like that. Let's talk a little bit about the disadvantage now. Is it okay? There's a lot of residual effect. There's a lot of other chemical reactions are going on because of the sodium hydroxide. Sodium hydroxide is a strong base. Bases catalyzes a lot of chemical reactions. You're studied in organic chemistry, right? Bases catalyzes a lot of chemical reactions, and that can essentially damage proteins. That can essentially bring new chemical reaction into play. And that's something I'm going to explain in the next couple of slides. Okay? The residual chemistry. One of the chemistry that brings the alkali extraction is, are you tired? Now we talk a little bit about chemistry, my favorite topic, and I'll take it in as slowly as possible. I have to rush again. I don't go in detail, but ask questions if it's not clear. Okay? Now we are talking about the residual chemistry that sodium hydroxide brings here. The first thing is that because of the sodium hydroxide, we create a new molecules in the system that's called lysinoalanine and lanthionine. We can also racemize the amino acids because of the alkali condition. Racemization is conversion of D to L form or L to D form. You know that the protein amino acids are always in L, but sometimes they can become D because of the OH minus ion. Okay? The first one is the lysinoalanine and lanthionine formation. Because of the OH minus, you break the disulfide bond here, okay? And you release a new molecule that's called dehydroalanine. Dehydroalanine, okay? And the OH minus can also take this SH group from cysteine as hydrogen sulfide. Actually, hydrogen sulfide smells like a fart, right? Basically, if you do a lot of alkali treatment, your protein can smell like rotten egg because of the hydrogen sulfide. And they release again the molecule called dihydroalanine. The serine residues can dehydrate, remove water molecule because of the OH minus, okay, and get the dihydroalanine. Now, all this reaction end up with the product called dihydroalanine, right? If you carefully look into the structure of dihydroalanine, it has C double bond C. Can you see? And the C double bond C is an electrophile, highly electrophilic in nature, right? Now, the protein contains nucleophiles. Can you see? Amino groups. And the thiol groups. Where is thiol group? Okay, it's missing in this structure. Okay, I'm showing its only product. For example, cysteine has a thiol group. Okay, lysine has an amino group, right? So, there is an electrophile here and there is a nucleophile here. Organic chemistry says what? There's a reaction. Electrophiles likes nucleophiles, nucleophiles likes electrophiles. They react and they make a covalent reaction. Basically, that will lead to the formation of protein cross-links. Now we have a protein with lysine here, and the protein with cysteine here, and the protein with dihydroalanine here. Basically, they react each other, and then you get the protein cross-links. A new cross-link is formed. Can you see? Because of alkali treatment. And please note that this is not a peptide bond. It's just a simple covalent bond here. Alright? Only peptide bonds can be cleaved in your stomach. Proteases can cleave peptide bonds. They can't cleave some other bonds. Now we created some other bonds. They can compromise the digestibility of the protein. Does that make sense? So because of the alkali treatment, we are creating the new structure. New structure means new functionality. That's a good thing or bad thing. But it can also compromise the digestibility of the protein. Make sense? It's one of the important chemical reactions often ignored because they don't know chemistry, this food science sometimes. They talk about functionality. So there is a chemistry is playing a role. Just to give some example here is that we have a method to analyze LAL and LAN formation in our department using LCMS. There's a paper I bought here. It clearly shows that increasing the LAL and LAN formation and then decreases here, right? So basically, you get more and more LAN formation depending on the pH of the solution. It decreases later because not because they're not producing. It's basically the LAL and LAN can also degrade further. Okay, so if you have high alkali condition, you can form more LAL, lysine-alanine, and also more LAN. This data is for LAL. Is it clear? And the same paper they published later the digestibility of that one. They show that the digestibility is also compromised because of the alkali treatment, because of the LAL and LAN formation. There's a compromised digestibility. Pepsin and trypsin cannot recognize LAL and LAN, and they can compromise the digestibility. And they also published, this is Chinese people, they published again showing that in the pepsin, this is pepsin, and this is pepsin plus trypsin treatment. This is pepsin. They clearly show that the release of the LAL is only 8%. Can you see? That means that the proteases cannot release them from the protein. If you have a cross-linked proteins made up of LAL and let's say LAN, if you put protease enzyme into play, for example, pepsin and trypsin, they can't cleave this one. They can't cleave the peptides here. They can't be released significantly. In some cases, only like less than 1%. And that actually compromises the digestibility. And nobody knows if you eat LAL and LAN, what happens to your body. It's a new molecule. It's not an amino acid. It's an amino acid, but it's not a normal 20 amino acids we have. It's a new structure. If you continuously eat LAL and LAN, I have no idea what is the health effect of that one. Less investigated. Is it clear? And the other thing is the D-amino acid formation. This is the most common form of amino acid in our food, Al. Okay? Because of OH minus, basically you remove this proton as water molecule, right? Chemistry. Then you get a carbanion formation here. So now if you consider it as a plane, so you remove, there was a hydrogen atom in this direction, and you remove that hydrogen atom, right? As a proton. Then the new proton coming can actually, now you lose the chirality, the new proton can come in this direction and also come in this other direction, right? And that will check the enantiomer, the racemization will happen. Now the L can become D. Please note that the D amino acids are different than the L amino. They look very similar, same molecular weight, everything is the same, but their functionality is different because our enzymes in our body apparently cannot recognize when there is a D. The bioavailability or the digestibility are compromised. And that some cases, they are actually digested. They also go to the bloodstream, and then inside your body, again, the protein synthesis should occur, right? And then there may be mismatching happens, and the functionality of the enzymes, muscle protein, can be compromised. Is it clear? So we really have to avoid D. The D is very less investigated. So L, the body likes the L. The D, they might be not digestible at all. They might be digested. They might be bioavailable. They might also do the protein biosynthesis inside, but the functionality of that new protein in your body may be a different one. So it's not a good molecule to have. However, there is no studies on this one. I could not find any single article, and we're going to publish one soon. Okay? Coming back to the color change. Whenever you do the alkali extraction, you get this color. Have you seen it? Red, brown, whatnot, all this different color you get on alkali extraction. And the product of the alkali treated protein is like this one. Brown, green, yellowish, whatnot, different color you get. It's simply because of the chemical reactions. Okay? In the previous stage here, I taught you that in this step, you actually precipitate the unwanted thing, and the supernatant contains the protein, right? It's not just protein, it also contains polyphenols, it also contains tannins, it also contains vitamins, whatnot, everything is present here, right? You're also up-concentrating unwanted things, and polyphenols, tannins, anthocyanins, they have color. So, it's a pure protein solution, we say, but not necessarily pure protein. It contains protein plus PP, polyphenols, tannins, whatnot. And if you dry it, freeze dry it, you get this one. You spray dry it, you get this one. Because of this small amount of 1% of polyphenols, less than 1%. Why they become color? I have a chemistry here. This is the structure of a polyphenol. It has OH groups. Can you see? They are called polyphenols because they have aromatic ring and hydroxyl groups. When they have a catechol structure, the catechol is basically the one with this neighboring hydroxyl group, the catechol structure. They are readily oxidizable. You don't need sometimes an enzyme, just pH can induce auto-oxidation. Okay? You have alkali condition, and that will induce auto-oxidation of polyphenol, and the polyphenol becomes quinones. Can you see? So this structure became this structure, quinones. The quinones are colored. They're highly colored. Okay? Alkali conditions favors this reaction, and that's the reason why you get this color. Polyphenols themselves may not be very colored. It's the quinone forms of the polyphenols are colored, and the quinone form is actually favored by the alkali condition, and that is called polyphenol auto-oxidation. But the reaction does not stop here. The quinone forms are highly electrophilic in their nature. Electrophilic because you have a carbonyl group. Can you see? Carbonyls, you have studied that in your organic chemistry. They are highly electrophilic. Okay? And the proteins have nucleophilic groups, amines and thiols. Electrophiles loves nucleophiles. And they just react together to two pathways, Michael addition or condensation, shift-based formation, addition reaction or condensation reaction from protein polyphenol adduct, a covalent compounds. Now the protein what you have here are bound to polyphenols. Right? Now it's not a pure protein any longer. It's a protein bound to polyphenol. It's called protein polyphenol adduct. And their surface properties are now different. Because originally the protein had cysteine group and lysine group, right? Now I have a polyphenol attached to that one. Can you see? Now the functionality, the solubility of this protein is entirely different because the polyphenol brings the OH group on top of them. Makes sense? Sometimes the solubility increases, sometimes solubility decreases because of the extraction, could be because of this reason. Because they are reacting with the polyphenols. And not only that, the digestibility of this one could be different. Because enzymes might love them or enzymes might hate them because polyphenols non-covalently can also interact with the enzymes because of their aromatic benzene ring. Because of their hydroxyl group, they can make hydrogen bonding. They change the chemistry completely now, so they can compromise the digestibility. They might improve, they might decrease. There's a lot of studies on that one. Is it clear why you get the color? Basically, this product is actually colored. If there's a shift-base formation on the protein, they're colored. Have you seen sunflower proteins? It's green in color, right? Right? Because of chlorogenic acid. And the protein reaction with chlorogenic acid gives a green adduct. So, sunflower extraction always get to green color protein because of this reaction. Does that make sense? So, conclusion here is that alkali treatment can oxidize polyphenols to quinones, and quinones themselves are colored. The reaction will not stop there. Quinones are highly electrophilic in nature. They react with the nucleophilic residues of the protein, lead to protein polyphenol adducts. And they themselves could also be colored, plus they can improve or decrease the protein solubility. They can increase or decrease protein digestibility because they are a very new compound now. Did I answer your question? If this reaction is done in only small amount of 1% polyphenol I'm taking. Protein is a high molecular weight. Polyphenol is small, but it's a low molecular weight, but still there's a lot of polyphenols present. Reaction can happen easily. So, this is the problem with the alkali conditions. Conclusion, coming back to your own questions, where do we get, what kind of yield you get? Here is some data. It's not a true one, I believe. It varies from the samples to sample, protein to protein, matrix to matrix, but this is something you can get it. Is it clear? Okay, let's try to conclude the extraction methods. Is there somebody to dare to conclude for me? I'm tired.
First, we start with the extraction method, that is conventional method. In that we can go with the dry and wet method.
Yes.
Right. In dry method, we go for the milling.
Yes.
We have to convert the cake form into flour form, or the whole form pea form into flour form, and then we go for the either air classification or the isoelectric charge separation. Right. So after that, then we go for the extraction via the solvents. And in those solvents, we use Osborne fractionation method. Either we can go for the salt solutions extraction, or either we can go for the alkali solubilization method.
Yes.
And they both have their advantages and disadvantages. And to conclude, means after that, do I have to tell about the advantages or
of course.
disadvantages?
Please go on. Yeah, it's your time now. Yeah.
Okay.
Yeah.
In salt one, we need to go with the determined level of the NaCl concentration.
Yeah.
And there might be chances of the residual of NaCN.
Yeah.
We have to go for the further purification as well, in case we want to have the maximum concentration of the protein content.
Absolutely.
Right. And when we go with the alkali,
salt concentration.
Alkali because in case of acid and alkali comparison extraction method, the yield is higher and alkali is cheaply available.
Yes.
So that's why we prefer the alkali extraction method.
Yeah.
And after that, the negative point of the alkali extraction method is that it may cause some unwanted reactions.
Yes.
Which may result in the formation of unwanted compounds, the consumption of which is unknown to the health.
Exactly.
Human health.
Exactly.
And the overall means extraction yield is here. And to conclude that alkali extraction method is best one preferred in case of conventional extraction method.
Yes. Well done. Very good conclusion. Claps for her. Yes. If you want to add more conclusion on top of that, yeah.
I want to add one more thing, that she told that alkali extraction is better than other conventional methods.
In terms of yield.
Yeah, in terms of yield.
Yeah.
But as per this health perspective, we should not use alkali extraction methods unless there is complete clear vision about the components present inside the
Exactly. The second part is very important. Unless you do not know. That's very important. I don't say that alkali is bad. Don't take that as a take-home message. Everybody is using alkali extra. It may be very good. Unless you do not know the other unwanted consequences, it might be.
As the compounds goes into the bloodstreams, they can contribute to the biomagnification, which is one of the main reason for different cancers and all these things.
Exactly. Food chemical toxicology. Yeah.
And one more point I want to add here, that in case of alkali extraction method, it will be always deficient in essential amino acids like lysine.
Yes.
Lysine is attaching with some
I forgot to mention that point. Exactly. Yeah.
Available lysine will be always deficient in these.
Absolutely. Thank you so much for bringing this point. Yeah. So one point I forgot to mention, and he is clarifying it. I told that this lysine is reacting here, right? Lysine is an essential amino acid, and many plant-based foods contains low amount of lysines. And I'm reacting them it further now. Now the available lysine is further reducing, right? It's actually a problem. Good point. Thanks for bringing that up. Yeah. Good. Any other points did we miss? Yes, she has some points, conclusions. Yeah.
I have a question.
Yeah.
Sir, when we are investigating some new protein.
Yeah.
And we would like to scale it up for the global
level. Yeah.
So in that case, what should be our primary point of interest? Whether the yield or the purity or the functionality?
Yes.
Among these three, which plays a major role?
We're all fighting for this one. I don't have an answer for this question. So, the conclusion here is that there is no protein that the method gives the highest yield, highest purity, highest functionality, highest nutritional benefit, highest health effect. No, it's a balance. It's a balance. It's a struggle that we are having today in this plant-based world. Okay? Well, how do we do this one? Usually, food industries talk about yield. For them, yield is very important because the yield brings money, right? If you talk into industrial perspective, yield is very important. Purity is also important. But usually, yield and purity go in opposite direction. If you want highest yield, you get lowest purity. Why? Because to get highest purity, you need to add another experiment. Membrane filtration, dialysis, ultrafiltration, column chromatography, all these new experiments should be added. Every single step you introduce in protein processing, you compromise the yield. Something is lost. 5% is lost, 10% is lost. Every step, something is lost. You're getting higher and higher purity, protein loss is also increasing. So it's a battle. Yield is the primary governing factor. I agree with, because yield is important. Functionality is also, because some industries produce a big batch of protein with highest yield, highest purity, and they sell it to you. After spray drying, after alkali extraction, you come to know that, oh my God, I have a lot, but it's useless. Right? And then you say, oh, it's highest yield, highest water holding capacity, I can make a meat analog out of it, I'm very happy, I'm selling it to the consumer. Consumer eats it, and then the nutrition specialist comes into play, and he says that, oh my God, it's very functional, but the digestibility is only 70%. Okay, then you're unhappy. Then the digestibility is 100%. Everybody's happy. Oh, it's very digestible, going into the stomach, going into the bloodstream. And then this guy comes, he's a health specialist, and he says, no, it has high amount of lal and lan also. And there is some study shows that lal and lan is bad for your health. I don't know it. It's not proven. Okay, there is a health concern. Where do we stop? It's again an iterative process. We always circling in the loop, right? At the end of the day, you need good yield, you need good digestibility and good health effect. It's just a loop. Research and development is required in this area. Nothing is the best. But I'm telling you that this unwanted thing could be maybe 5% is a problem. No, 95% is. I don't say that every protein is damaged. 95% may be still safe to eat. Only 5% proteins are damaged. It's more about how much you eat every day. If you continuously eat damaged protein, maybe you have a health effect. If you eat once or twice in a day, just excrete it, because our body has a fantastic defense mechanism to excrete unwanted molecules. That's the beauty of human being. That's the beauty of animals. The problem, the health issue only occurs if you continuously consume unwanted things, usually. They're not poisons yet, right? The poison is also depending on how much you drink, right? If you eat small amounts of the poison, nothing will happen, right? Smoking, alcohol, nothing will happen. Continuously eat, you get your liver damage. If you eat one bottle of poison, you might die, right? It depends, the minimum dosage, the L350. I'm not a health scientist, but I know these things, but it depends. Did I answer your question properly? I don't have a right answer. It's a debate. Yeah.
And one more thing. The protein polyphenol adducts that are formed, is it possible to be removed by membrane filtration?
Super good question. That's whole, we spent 3, 5, 4, 7, 6 years of work on this one. It's impossible to break this bond. We are not successful yet. There is no enzyme, at least we have identified, that can cleave this one. So if they're covalently bound, it's very difficult to remove. It's permanent. We could not identify any enzyme or chemical treatment that can efficiently break down. We can use some reducing agent, yes, but as soon as you add a reducing agent and oxidizing agent, you also damage the proteins. Yeah. We don't want to damage protein. We just want to get rid of this reaction compounds. It's very difficult because food is such a sensitive molecule. We are not using it for the bioplastics. We're not using it for because soy protein can also use to prepare gum, glue. There you can do anything you want. Nobody eats the glue, right? But food, we want to eat it. If you want to eat it, you have to be careful. I can do some chemical reaction to break this bond, but you can also break the proteins along the same way. It's a tricky situation.
Is there any method to identify?
Yes, we have a lot of LCMS methods. You can use untargeted and targeted methods. That's one of the expertise of our own labs. I do that research together with my colleagues and students. Okay. There are methods, yeah. I did not cover all these things at the interest of time, but we can talk about it later. Yeah.
I want to answer her question. These phenolics can be easily removed. Before going for the extraction, we can dephenolize the meal.
Yes.
In which these phenolics are present. We can dephenolize the meal. After that, we go for the extraction.
I agree with you and I partially disagree with you also, because this is something we have done it so many times. We can actually dephenolize somewhere in this particular step, right? We can remove as much phenolics as possible. But it's impossible to remove every single phenolic compounds, because as soon as you bring the protein into solvent, a solution, as soon as you crush the seeds, phenolics actually located in one particular region of the seeds. They're not present everywhere inside the seed. But as soon as you crush them, they interact them. As soon as you put it into solution, they're very happy. Now I can move everywhere, right? They can just attack proteins, whatnot. And there is a problem. It's very important to do, when do you dephenolize, right? It's impossible to dephenolize when the phenolics are covalently reacted with the protein. You can dephenolize free, non-bound phenolics, not the bound phenolics today. And this is something we are doing it with our rapeseed project. We are doing tons of experiment. In fact, it's actually the rapeseed protein powder. I should not tell it. Okay, I'm on a video. The rapeseed protein isolates actually look like this. We did a lot of dephenolization, we did a lot of purification, blah, 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 whatnot. We still have color. Tell me from where it's coming. Yeah. We have tried everything one could try to get the color out. It's not possible. Probably because of this we don't have a proof for this. It has happened. Probably because of this covalently reacted compound. They are difficult to remove. The small amount of phenolic, they are present somehow. It's impossible to remove. Yeah. Or less possible, less easy, it's very difficult. I don't say impossible. Impossible is a bad word to use in science. So difficult is the right word, maybe. Yeah.
I have a suggestion.
Yeah.
So if you have used a lower molar NaOH, based on my experience, I'm sharing it with you. If the molarity of NaOH is less, then you get a more less colored protein.
Yes, exactly. Yeah.
And also the solid to solvent ratio, that also plays a major role. Because if you are using 1:20 ratio, you have to add more NaOH, which gives more color to your protein.
Exactly.
So
that's where you play around. Again, there you've come into the yield battle now. So then you compromise yield, because high alkali condition, more yield, more color. Low alkali condition, less color. Yes, yield is also compromised, but the industry wants yield, right? It's a battle. So No way.